Hi, my name is Lindsey Giffey and I'm from WF West High School. For my research project, I was trying to determine the effect of the duration of light regarding desynchronization on the period gene in Dorfacillomylogaster. And in this project, I was simulating a flight from Seattle, Washington to Athens, Greece to see if there was a light pattern that would best adjust fruit flies to the new time zone. So desynchronization is caused when a person is not adjusted to the time zone that they are currently in. This happens when you change time zones quickly and is commonly known as jet lag. This includes side effects such as fatigue and headaches, which are a nuisance to travelers. Desynchronization is caused because the circadian rhythms of the person who traveled are not in sync with the new time zone. Circadian rhythms are the body's natural clock, and they control time-based functions. That's why people tend to wake up a couple minutes before their alarm clock, or they get hungry at the same time of day because their body is telling them that they should be hungry or they need to wake up soon. This is caused because of the protein levels of the period and timeless genes, which control circadian rhythms. The timeless gene is one of the genes that regulates circadian rhythms by the protein that it produces. This protein is light sensitive, so it is not stable during the day, but at night is able to build up in the cells. The period gene is the other main gene involved in regulating circadian rhythms. In order for this protein to be stable in the cell, it needs to form a complex with the timeless protein, but since the timeless protein is being broken down during the day, it cannot form a complex until night, so the protein does not build up during the day, but it is built up at night. These two genes regulate their own mRNA production based on the amount of protein that is in the cells. At night, once there's enough of the protein complexes built up in the cell, they move into the nucleus and turn off the production of their own mRNA. This is caused when they form a supercomplex with the clock and cycle proteins. Once this supercomplex is made, the mRNA is no longer produced. When light is reintroduced in the cell, the timeless protein is broken down, the supercomplex is broken, and mRNA is produced in the cells again. Here we have a diagram of what is occurring with the period and timeless proteins. So in the morning, the genes are turned on, so the mRNA production of the period and timeless protein is occurring, which causes proteins to be produced. During this time, light is breaking down the timeless protein, while kinase breaks down the period protein. At night, when light is taken out, the complex forms and moves into the nucleus to turn off the genes by forming a supercomplex with clock and cycle complex. Light is re-entered into the cell, and the gene is turned off. And that cycle just keeps going every day. For this research, it's hypothesized that on a simulated flight between Seattle, Washington to Athens, Greece, that the fruit flies that were given light between 3 p.m. to 7 p.m. Pacific Standard Time would be best adjusted to the Athens time zone, because they would be waking up slightly before light would be entered into Athens, which would allow the complexes to break, causing the mRNA to be produced again, and this way the cycles would get synced up. For this research, I had 7 different groups. There was a Seattle control and the Athens control, and there was a all light exposure control and a no light exposure control, and then there were 3 different test groups that were given no light at varying times throughout the flight. To begin this experiment, the flies were separated into 7 groups that had 3 trials each. The 6 of those 7 groups were adjusted to Pacific Standard Time, and one of the groups was set 10 hours ahead to Athens, Greece time for 1 week. After the week, a simulated flight occurred from 11 a.m. to 10:30 p.m. Pacific Standard Time. Immediately after the flight, the flies were euthanized, and then the RNA-Nucleus 4 PCR kit from Life Technologies was used to extract the mRNA from the samples. This kit consists of destructing the cells and then putting the sample through a spin column with several wash solutions in order to separate the DNA. The extracted mRNA was then converted to cDNA using the Taqman reverse transcription reagents kit from Life Technologies, and this was run on the PCR machine where it was a one-cycle process where the mRNA was converted to cDNA. After this was completed, I created two master mixes for the real-time PCR reaction. One of them was the 18S control, and the other was the period gene custom assay. The 18S control was used for this experiment because the 18S mRNA and protein is abundant in all cells in fruit flies, and this was a good way to make sure that the mRNA was extracted right and the cDNA and converted to cDNA correctly, and that the reactions were set up correctly. After these master mixes were made, I loaded and ran a plate on the real-time PCR machine on the recommended settings. Here we have several pictures of me working on my experiment. In the left two pictures, I am using FlyNap in order to, as an anesthetic, in order to move the flies from between vials more easily. It is a common anesthetic used to move flies. In the right picture, I am using the RNA aqueous 4 PCR kit in order to extract the mRNA from the cells. Here's a picture of the amplification graph received from the real-time PCR reaction. The blue and purple lines are the 18S control, and the green, red, and yellow lines are from the period gene assay. It was expected that the 18S protein or 18S mRNA would amplify more quickly and reach the CT line first because there is a lot more of that mRNA present in the cells of the fruit fly. For analysis, the mRNA extraction and the reverse transcription process were successful because they both had high concentrations when checked on the nanodrop, which is a machine that we have that accurately checks how much DNA is present in a solution. Because of these high concentrations, it proved that the RNA extraction was completed correctly and that the conversion to cDNA ran well. Since amplification occurred, the real-time PCR reaction was successful. The 18S controls reached the CT line by cycle 25, and the period mRNA reached the CT line by cycle 32. This showed that there were moderate amounts of cDNA in the reactions, but based on when they reached the CT line. The results also showed that there was 10 to 12 times as much of the 18S mRNA in the flies than the period mRNA, which was expected since there is a lot more of the 18S mRNA present in the cells. Here's a graph of the relative quantification of the reactions. These were compared to the no-light control 1. For this, the samples are compared to the no-light control in regards to when they reach the CT line. They also take into consideration when the 18S control of that same reaction reached the CT line to account for slightly varying amounts of cDNA in the reactions. This way you can tell which samples are most closely related compared to the one sample that you choose as the sample that you look at. Here's the relative quantification graph converted into a table. Here we have the relative quantification mean for each of the trials and the standard deviation in comparison to that. You can see that the Athens control and the no-light control and the no-light from 3 to 7 have similar values on this table. For analysis of the relative quantification, the Athens control was most similar to the no-light control of all the test groups. Of the 3 variable groups, the Athens control was most similar to the 3 p.m. to 7 p.m. test that had no light between 3 p.m. to 7 p.m. For all of these, the standard deviation was fairly low, and there were 2 groups that were above 1, but neither of these groups were ones that were very similar to the Athens control. Here's a table of p-values received from these results. There are several that are very low, and that concludes that they have no similarity to the Athens control. These include the Seattle control and the no light from 7 p.m. to 6:30 p.m., which proves that they have no similarity to the Athens control. The no light control, which had a p-value of 0.606, was the highest of all the p-values, and although that does not mean that they are extremely similar, it shows that there is some similarity between the two. Of the variable groups, the no light from 3 p.m. to 7 p.m. had a p-value of 0.264, which, although is still very low, this variable had the most correlation to the Athens control. So based off the p-values of the variable groups, the 3 p.m. to 7 p.m. with no light was the test group that was the closest to Athens control. But of all of them, the no light control was the closest to the Athens control. Neither of these values are great enough to draw any finite conclusions, though. The no light would theoretically help the person best adjust to Athens based off the data given, but the person would not be fully adjusted. For conclusions, the RNA was extracted successfully because there were high concentrations of RNA when checked on after the extraction, and the real-time PCR reaction worked. And if the RNA had not been extracted correctly, then the real-time PCR reaction would not have worked. And the cDNA conversion was also successful for the same reasons as the RNA extraction. The results somewhat concur with the hypothesis because of the test groups. The 3 p.m. to 7 p.m. was the most beneficial of the three, but the no light control was unexpectedly more similar to Athens control than all the other groups. None of these were as similar to the Athens control as hoped for, though. And since the no light control was the closest, this shows the importance of extended periods of darkness throughout the flight, which would be useful information on future research. For future research, I would increase the no light period on the flight, but I would also take into consideration that waking them up around 7 p.m. Pacific Standard Time helped adjust them better based off of those two values since they were the highest. And I would increase the amount of time that they were given no light, and I would also vary the time that they were woken up at that time. I would like to acknowledge my teacher, Henry Weeks, for being very supportive and helping me a lot through this research project. I'd also like to thank David Kogan from Life Technologies for allowing us to get labs equipment and supplies at a discounted rate, and I'd like to also thank my parents for being supportive through this process.